Up with week 7, we're going to be talking more about the immune system, continuing with chapter 21. And so, like we talked about last week, the immune system relies on things like the lymphatic system and the circulatory system to actually have a physical anatomy, a physical location in the body to exist. It's much more of a conceptual system. We talked about the first and second lines of defense being part of the innate immune system that recognizes generalized problems. So things like it prevents things from getting inside of the skin. So most things stay out, most things inside our body can stay inside. It also helps with generalized problems like natural killer cells that recognize cancer cells that have gone awry, or cells that are infected with a virus that send out those interferon signals saying we're having stress, there's a problem here. It also includes things like the macrophages and the dendritic cells and the neutrophils that are phagocytes that gobble up foreign stuff and recycle those contents. And it just generally recognizes something as not being self, not being accurate, not being correctly functioning, or not being in the right place at the right time, not being the right cell type. So it doesn't know specifically that it's a Staphylococcus aureus bacteria, or specifically that it's a tuberculosis bacilli bacteria. Instead, it just recognizes it's foreign and shouldn't be here. Very often, though, our body can't rely strictly on those first and second lines of defense. The second line of defense will only go so far. A fever only generally helps slow metabolism of the bacteria and increase some of our metabolic activities. Okay, so humans and other different advanced animal species that have evolved much more complex immune systems have an adaptive or a specific immune system. So when we talk about the adaptive immune system, we're thinking specific defenses that eliminate almost any pathogen or abnormal body cell, provided it has time to figure out what actually is foreign. Okay, so it amplifies the inflammatory response. It activates complement. However, it must be primed. It must have time to figure out what the problem is. So it needs an initial exposure to that specific foreign substance, and that priming will take time. So we don't get an adaptive immune response right away. That's why very often infections take maybe a week or more to get over if it's a severe infection, because your body takes time to kick in that adaptive immune system if that's what's needed to cause us to actually get over whatever the sickness or illness is. So some characteristics of the adaptive immune system: it's always specific. Okay, so it doesn't say, oh, this person looks like a dangerous threat because it has a gun going through the metal detector. Instead, it recognizes the specific look of its face, or recognizes the fingerprints. Okay, so translate that into the immune system. It recognizes a specific antigen, that shape that it knows has been a problem before, and tries to prevent it from being a problem in the future. It's systemic, so it's not restricted to the initial site of infection. So if we get a cut on our finger and Staphylococcus aureus leaks inside, it's not going to limit the response to the finger. It's going to be anywhere throughout the entire body, anywhere in the blood system and the lymphatic system that potentially might have that physical shape, that antigen, aka anywhere that has that bacteria. And then finally, it has memory. This is what allows it to be more effective. Once it knows what the antigen is, the physical shape that it can recognize as foreign, it mounts an even stronger attack the next time. Okay, so we call that the second response or secondary response, and we'll talk about that more later. So the two main branches within the adaptive immune system basically fall into B cells and T cells. Okay, and we call those humoral immunity or antibody-mediated immunity, and cellular immunity or cell-mediated. And in humoral immunity, we're really talking about the bloodstream. So remember, in older medical texts, we used to call blood humors. So humoral just means in the blood. So humoral immunity or antibody-mediated immunity is actually going to be where B cells mature into plasma cells, and those plasma cells produce lots and lots and lots of antibodies to recognize something as foreign and tag it to make it more effective for the phagocytes and for complement and for some of those other systems to be able to attack and get rid of the foreign invader. The cellular or cell-mediated immunity is actually going to be T cells, and those T cells specifically are going to attack whatever the foreign substance is. So humoral immunity, B cells make antibodies that basically kind of tag something as needing to get rid of and make it easier for the body to eliminate it. The cellular immunity in the T cells is actually itself going to attack the foreign invader. And that's an oversimplification, but for the purposes of this course, that's a very good way to think about humoral and cellular immunity and the functions of B cells and T cells. Okay, so humoral immunity, we have antibodies that are produced by those B lymphocytes circulating freely in body fluids, like blood, like the lymphatics. They bind temporarily to a target cell, they temporarily inactivate it, and mark it for destruction by phagocytes or complement. And humoral immunity has various extracellular targets. It recognizes specific shapes on the outside of foreign invaders. Cellular immunity is where those T lymphocytes act against a target cell. They either directly kill the infected cell or indirectly by releasing chemicals that help to increase inflammation or activate other lymphocytes and macrophages. So cellular immunity has specific cellular targets. B cells and humoral immunity is outside extracellular targets. T cells and cellular immunity is the cell itself that it's recognizing. So this brings up the term antigen. We've mentioned it in last week, but let's actually define it and talk about some details of antigens. So antigens are any physical shape, any chemical substance that can mobilize the adaptive defenses and provoke an immune response. So an antigen can be a self-antigen, or it can be an antigen from a foreign substance. So it's a way of basically recognizing, is it me or is it not me? And specifically, is it something else? So there's me antigens or self-antigens that recognize a liver cell as being important and functioning properly, it's part of my body, or recognize a nervous system cell as part of my body. And then the immune system also has the ability to recognize antigens as a specific foreign invader. Okay? So the target of all adaptive immune responses is actually the antigen, the physical shape that it can recognize. Most are very large, complex molecules, not normally found inside of our body. They're non-self. Characteristics of antigens, they can be complete or incomplete. We call those haptens. We'll define that in a second. They can contain what we call antigenic determinants, and they can be a self-antigen, like I mentioned briefly. So antigens can be complete or incomplete. Complete antigens have two important functional properties, reactivity and immunogenicity. What do we mean by that? Immunogenicity, they can actually stimulate the proliferation of specific lymphocytes. They make lots and lots and lots of B and T cells grow because they're trying to fight off that specific antigen, that specific physical substance that it recognizes. Reactivity, meaning that actually those lymphocytes that are going to be proliferating can actually themselves attack and bind to that antigen and eliminate something. So, for example, foreign proteins like polysaccharides, foreign proteins as well as polysaccharides, lipids, nucleic acids, all may be seen on the outside of foreign invaders or the outside of pollens or various toxins, and it recognizes that physical shape. It causes lots of lymphocytes to be developed really quickly to be able to attack specifically the antigen. So that's immunogenicity and reactivity. Incomplete antigens or haptens involve molecules that are actually physically too small to be seen and are not themselves antigenic, but can combine with other stuff to become an antigen. Okay, so really small peptides, small pieces of proteins, really small individual nucleotides, and certain really small hormones. So they only become immunogenic if they attach to specific proteins in the body. So, for example, the protein and the hapten, like the foreign hormone, combine together, and the body now recognizes the physical shape of them bound together. Okay, so it's really pretty simple. Most things we think about are themselves complete antigens. Some things, though, only trigger an immune response, an adaptive immune response, when they bound with something inside of the body, like a protein. Causes immune basically a complete antigen and haptens. They can cause the immune system to mount an attack that is harmful to a person because it attacks self-proteins as well as haptens. Okay, so some examples would be poison ivy. All right, people that have problems with animal dander, different detergents, and some cosmetics. So antigenic determinants are parts of the antigen that antibodies or lymphocyte receptors actually physically bind to. So somewhere that once it recognizes the shape, it can physically hook on, kind of like the handcuffs that hold it in place. Most naturally occurring antigens have lots of antigenic determinants, places that can actually be hooked onto by antibodies or by the T lymphocyte. And they mobilize several different lymphocyte populations. They form different kinds of antibodies against them. We're not going to talk a lot about the different types of antibodies in this class, but we will mention them briefly. And the antigenic determinants are going to be large, chemically simple molecules, even things like plastics, that have little or no immunogenicity. Okay, so there are large chemical substances that are very simple repeating substances that don't actually cause an immune response. So here's a picture of an antigen, all right? And it can have lots of different places on its physical shape that different immune cells, different lymphocytes recognize. So for example, the B cell over here that makes antibody B recognizes this kind of rounded shape. So it makes antibodies that specifically can bind to that physical shape. On the other side of the antigen, there may be another physical shape that's recognized, kind of this kind of block castle shape, that's recognized by the B cell over here that makes antibody C. Okay, and then a third type of lymphocyte might recognize this physical shape and make this antibody that specifically binds to it here. So the antigenic determinant is the physical shape where the antibody can bind into after it's been recognized by the B cell or the T cell. So I mentioned already that we have self-antigens. This is very important. So ways of determining that this cell is in fact a normal healthy cell of our body. So self-antigens, all cells are covered with a variety of proteins located on the surface that are not antigenic to self, but may be antigenic to others when we have transfusions or grafts. So for example, an organ transplant. If I give someone one of my kidneys, their body is going to see many of the physical shapes on the kidney and on the cells, on the tissues, on the proteins as being a foreign substance. And potentially their immune system will react to it. Okay? So one set of important self-proteins are glycoproteins, so they're sugar proteins that we call MHC proteins, which stands for major histocompatibility complex proteins. They're unique to every individual, but we can broadly group them together. So kind of think like blood types when we talked about the ABO blood grouping and the rhesus factors, and we said that there are lots of different proteins, and it's which proteins your body makes. And so if you match up, then potentially you can give blood to the same blood type, or someone who has have the same proteins or less proteins than you do. They can be a donor for you. So same idea here, except for we have different major proteins that exist on our body cells, and that kind of like potential self-antigens that other individuals would react to, but our body does not. Basically, these MHC proteins contain a groove that can hold a piece of self-antigen or a foreign antigen. So there's two different types. We're not really going to go into it right here. But one type of MHC proteins actually helps to tell the body not to react to self-antigens. And another type of MHC proteins actually helps to tell the T and B lymphocytes, hey, this is a very specific shape that you should recognize as foreign. Okay? If you're interested in that or have any interest in immunology, you might want to consider a career actually in histocompatibility. There are technologists that work in a research lab that deal heavily with organ transplant, and their job is to actually match donors and recipients based on their MHC typings. So it's very important in transplant medicine and several other areas of medicine as well. Okay, so the adaptive immune system involves three different cell types. Okay, the two types of lymphocytes, B and T lymphocytes. We already said B cells or B lymphocytes are going to be humoral or blood-based immunity. That's antibodies. And the T lymphocytes or T cells are cellular immunity. So they themselves are going to do the attacking or trigger other chemical responses and immune system responses. The antigen-presenting cells is a third type of cell we haven't specifically talked about. We've talked about examples, but we haven't talked about this specific role. So to be able to recognize a foreign substance as a specific foreign invader. We need to have some way of actually identifying the problem and passing it on to the B and T cells. And that's the job of the antigen-presenting cells. They pick up potential antigens from different things they see going through the lymphatic system or going through the blood system, and they show them to a B cell or a T cell and identify it as foreign. Okay, so the antigen-presenting cells, which we'll talk about more in a second, do not respond to a specific antigen, but basically help to provide antigens to the B and T cells to let them know what's in the body and to recognize specific threats. So you can kind of think about them being like the security cameras that present the images to the security guards, the B and T cells, that actually make the decision on which cells should be arrested. Okay, so lymphocytes develop and mature and have to be totally activated. And so we already said that T and B cells differ because the T cells mature in the thymus, the B cells mature in the bone marrow. Okay, both sets of cells actually start in the bone marrow, the red bone marrow. So here's kind of a picture. They both start in the red bone marrow. Okay, the T cells are going to go to the thymus. The B cells are going to stay in the red bone marrow to mature. So we sometimes say that they're educated in about a 2 to 3 day process in those primary lymphoid organs, the B cells in the bone marrow, the T cells in the thymus. The lymphocytes get educated for 2 reasons. Okay, self-tolerance, meaning we don't want to respond to our own antigens. We don't want to attack our own cells and recognize those physical shapes as foreign. So we have to make sure that those B cells and T cells do not attack our own antigens, our own physical shapes. And the second reason is to develop immunocompetence. Okay, that means they have to be able to recognize one and only one physical shape, only one antigen. So every B cell or every T cell that has matured in your body only recognizes one specific antigen. It recognizes this one shape that's part of Staphylococcus aureus, or this one shape that's part of the tuberculosis bacilli bacteria. Okay? So you may have B cells and T cells that recognize different antigens of the same pathogen. All right? But that's very important. The maturation and education process makes sure that we attack one and only one foreign antigen, and we don't attack any of our own antigens. So that's very important for both B and T cells. Okay, so maturation occurs either in the thymus or the red bone marrow. And then the seeding is what it's called, basically of secondary lymphoid organs. So once they're immunocompetent, once the B and T cells mature in the bone marrow and the thymus, they haven't actually seen a physical antigen yet. Okay, so we call those naive, meaning they haven't been exposed to anything yet. They go from the thymus or the bone marrow into all the different secondary lymphoid organs. So the lymph nodes, the spleen, anywhere that has any surveillance, the malt organs. This increases the chance that they actually encounter that specific antigen. Okay, so the seeding in the lymph node, for example. And then they actually find the antigen. They encounter the antigen, like during an infection. So naive lymphocytes first encounter the antigen, and that triggers the lymphocyte to develop further. Once it develops a little bit more, we go through what we call clonal selection. Basically, we're going to make lots and lots of the exact same cell that can only recognize that one antigen. And that's what's going to allow the T cell or the B cell to actually attack or make antibodies to attack the foreign substance. Okay, so the last step, the antigen actually encounters the antigen, and that activates it to make lots and lots of those either B cells or T cells. Okay, and that would be the step proliferation and differentiation. So those clones that are made, they're going to be made into effector cells, and we're not going to go through a lot of the different cell types. There's a lot of detail here, particularly with the T cells. There are different subsets of T cells that have slightly different functions. So they're not all doing the exact same thing in the exact same way to help combat potential pathogens. But I do want you to recognize that B cells, it's not the B cell itself that makes the antibody. Instead, the B cell, once it proliferates and begins to differentiate and become mature, it actually becomes what's called a plasma cell. We'll see that in a second. And then a few remaining B cells remain as memory cells. And they're going to, even after the infection is over, the plasma cells make lots and lots of antibodies and then die off as the infection's been destroyed or defeated. The memory cells are going to be B cells that sit around in those secondary lymphoid organs, and they remember that same physical shape. So the next time you get exposed to that same antigen, it's there to make a very quick response. We don't have to go through steps 1 through 4. We're ready to respond right away and proliferate and make more B cells and plasma cells to be able to make antibodies. Okay? And then there's also memory cells that are going to exist in the T cells after an infection. So you remember next time what things looked like, what the antigen looks like. All right. I've included a few slides about things like the receptor diversity of an antigen. So actually the antibodies recognize lots of different physical shapes. And it's a very detailed process, and it's actually very interesting. You might want to take some time to look at that or read about that from the book. But for the purpose of this course, we really don't have time to go into antibody structure and how we could actually recognize so many different shapes that potentially could exist in all the different bacteria or all the different types of foreign invaders. Okay? So you can read through these next few slides on your own time if you'd like to, but they're really beyond what we're going to cover in this particular course. All right, those antigen-presenting cells, okay? So those cells that actually are sitting around, typically phagocytes, gobbling up potential foreign substances. As they gobble them up, they're going to take specific proteins, specific fats, specific sugars, and present them to T cells and B cells for potential recognition. We call those antigen-presenting cells. And they're primarily dendritic cells and macrophages, or B cells that have already identified a specific antigen to attack, and they will all activate T cells and tell them this is a specific antigen. And the B cell and T cell would actually be able to attack and remember that one specific antigen. So dendritic cells, I already mentioned briefly, they actually look like they have dendrites. They have no connection to the dendrites of neurons, but physically they kind of look like that. And so dendritic cells and macrophages are very similar, and their job is essentially to present a potential antigen to the B cell and particularly to the T cell. All right. So B lymphocytes do not activate naive T cells, the ones that haven't seen an antigen yet. They present antigens to a similar set of T cells that help to activate them. We're not going to learn the specific subsets and types of T cells in this class, but you're welcome to. There's a lot of detail in your book, but it goes much more advanced even than that in immunology. So the humoral response, the B cell response with antibodies, okay? So once the B cell encounters a target antigen, it's going to provoke a humoral immune response. The antibodies specific for that particular antigen then get produced, okay? They recognize that physical shape and can attack it. The B cells are activated when the antigen binds to the surface receptor. We won't talk about the exact chemistry of how that happens. And that leads to proliferation and differentiation of the B cells. So you get more and more of them and the development of clones that we call plasma cells. So they're going to be circulating throughout plasma, and their sole job is to make antibodies to recognize that specific antigen. To secrete specific antibodies, it does that at a rate of about 2,000 molecules per second for 4 to 5 days. So think about how many physical antibodies, those little proteins it must be making. About 2,000 antibodies per second for 4 or 5 days. Okay, so it basically blankets your bloodstream and your lymphatics with lots of these antibodies that can recognize the foreign substance. The antibodies circulate and mark them for destruction by either the innate immune system or other aspects of the adaptive immune system like T cells. Okay? And in fact, in your first response, the first time you see an infection, it takes time to develop those first few steps until you make the plasma cells that can make antibodies. The next time, the secondary response, the next time you actually get exposed to the same antigen, in fact, you're going to have lots and lots of plasma cells that are made. So you can see here the example of 3 cells versus 1, 2, 3, 4, 5, 6, 7 cells that are made the second time you get exposed to that same antigen. It's also going to happen much more quickly. That's why you respond much better the second time you've been exposed to a potential infection or pathogen. So some of those clone cells that don't actually become plasma cells, that don't actually mature and make antibodies, so like this right here, will remain as memory B cells. So you remember for the next time you might get exposed to that that particular antigen. So the primary immune response, the first time you get an infection or the first time you see a specific pathogen, cell proliferation and differentiation, once you get exposed, you have about a 3 to 6 day period to actually get to the point where you're going to make antibodies and actually make mature T cells that can go out and attack things. Okay, 3 to 6 days. That's why very often if you can't take an antibiotic or a specific drug to combat that pathogen, you have a period of maybe about a week before you start feeling better. Peak levels of antibodies circulating through your plasma are actually around day 10, and gradually the antibody levels decline. So you're still combating the infection for several days beyond that. The next time you get exposed, or the secondary immune response, so let's say, for example, you have the flu, you get over it in about 6 or 8 days, and then 2 weeks later you get exposed to the exact same flu virus because you have a family member that gets sick, and you're helping to take care of them. When you get re-exposure to that same antigen, the same strain of the flu, your body's going to get a much faster and a much longer response that's going to be much stronger. Therefore, it's more effective. So the immunologic memory B and T cells are going to respond within hours instead of 3 to 6 days. Antibody levels will peak in 2 to 3 days, but in a much stronger level, and are actually better at binding with the antigen. And sometimes those levels remain high for weeks or months. So let's say this is like when you first get the flu, okay? Around day 10, you have the most antibodies in your system. See, it takes several days before you even first get a response. Once you get the specific adaptive immune system response, gradually you're going to get a lower level of antibodies. Okay, so in this example, let's say that you totally don't have any more antibodies floating through your bloodstream after about a month at day 28. But let's say that at this point you have a family member that gets exposed to the same flu strain. What's going to happen is your body, within a matter of hours, is going to start to create a much stronger response. So see, here it's 10 to the 1 antibodies. Okay, so about 10 antibodies per unit in the body. And then here, it's almost 10 to the 4th. So that's like adding 3 more zeros after the 10. Okay, so a much stronger response the second time. It happens much more quickly, and it's going to last for longer. So see, we're not even down to that strongest response level yet at day 56. That's why very often we never actually see symptoms and never actually get the infection from whatever the foreign invader is upon second exposure. So this brings us to different ways we can get immunity. We can have active or passive humoral immunity. So again, talking about B cells and antibodies. And it can come naturally or be artificially acquired. And in the lab, we've talked some about kind of the flu exercise and a little bit about how vaccines work. So kind of to expand on that, active humoral immunity is when your body actually gets exposed to something, develops its own immunologic memory, and then is able to attack on its own the next time you get exposed. So the most common example would be naturally acquired active humoral immunity. So naturally acquired means you get the infection. For example, you get chicken pox when you're 7. Okay, you go through the process of getting over the disease, and your body develops memory of B and T cells. And then the next time you're exposed, you're ready to respond quickly and much more strongly. All right. Artificially acquired would be something like you get a vaccine. We give you a little bit of that chickenpox virus, okay, in the form of a vaccine. It's a dead or attenuated pathogen that can't cause the disease, but your body can get exposed to those physical shapes and develop its own recognition of those antigens. So it knows the shapes and develops its own B and T cells. So when you actually do get exposed to the pathogen, you're able to create your own antibodies and your own specific T cells that recognize that antigen. So active means your body itself makes the antibodies and will make the T cells to attack it, whether it's naturally acquired or made artificially. Passive immunity occurs when antibodies that are already made or developed are introduced directly into the body. So for example, B cells are not challenged by the antigens themselves, so the body doesn't make its own memory. Okay? So as long as we give you the antibodies, you're fine and you're protected, but as soon as the natural levels of the antibodies go down, your body degrades and recycles the proteins of the antibodies, you're no longer protected. Okay? So it's a temporary protection. Probably the best example we think about this is naturally acquired passive immunity. And so this happens before birth when the fetus and the mom are still connected, their bloodstreams are connected by the umbilical cord. And so what actually happens via the placenta, mom's antibodies get passed into the fetus. So if either one of them get exposed to an infection, potential pathogen, her immune system is protecting the fetus. Additionally, this happens during breastfeeding. Okay, so one advantage of breastfeeding over formula is actually the baby will get some natural antibodies that mom's body might make to give on to the infant. So let's say that 5-year-old brother comes down with a chicken pox. Well, the infant might actually be protected if mom is breastfeeding, because if she's already been exposed to the chicken pox, her body starts making some antibodies that may get passed along to the child and protect the infant. Okay? The infant can still get chickenpox later because they don't develop their own memory cells. Okay, so once breastfeeding stops, they're no longer protected. This can also be artificially acquired. So, for example, if we actually inject the serum of somebody that has the antibodies, so maybe you've been exposed to a specific virus before, okay, maybe it's a rare virus, and got through the infection. If you have the same blood type, maybe experimentally they're worried about a patient. Maybe they actually give a little bit of blood, of your blood, to that patient, and hopefully that'll help them to overcome the infection themselves. Hey, I'm coming.
The door's locked. I'm so sorry. Did you finish? Yeah, how that thing out?
Oh, that wasn't on purpose.
Sorry. If that thing's open too. Oh, it's hot. Sorry.
You're good.
She said, "What's he doing? Talking to somebody on the phone on some Andy's." I was like, "Oh, he's tutoring." And I was like,
No, I'm
he's
recording.
the
That's fine.
videos.
That's fine. How did it go? Okay, I kind of was glancing. I'm like, are y'all still? It's like I figured y'all were still doing something.
We're trying to find someone to sign our papers.
Oh, part.